Good evening, everyone, and welcome to Genomics Lite. Thank you, everyone, for joining us for our second session in our cancer research series. So tonight we're joined by Patricia, Jaaks, and Luke Harvey, and we'll be looking at how we use genomics and how we use lab skills to help us understand cancers and to treat them more effectively. So we're going to look at two halves of the story tonight. So Luke's going to talk us through how we use healthy samples, so how we study cancer before it's happened and how it happens and evolves. And then Patricia's going to help us look at what we do when we do have a cancer, how we can take a tumour and hopefully understand it and find a drug suitable for working towards getting rid of it. So I'm going to share my screen again quickly and talk you through how the session's going to work. All of our sessions are aimed to help everyone explore what genomics means for our lives, and we ask everyone to join us in a spirit of curiosity in our sessions. Our sessions are being moderated by myself and my colleague Fran tonight to make sure it's a safe and inclusive space for everybody. If you've not used webinar before, you've probably spotted that this you can't use your camera or your microphone this evening. Instead, you can use the chat box and the Q&A to get hold of us. If you are having any technical issues or you want to say hi, use the chat box at the bottom of the screen below. During the session, our panelists might ask you a question. They might want to see what you think about something, so use the chat box to reply to them as well. If you have a question for either of our speakers this evening, we'll have time for questions in between and at the end. So pop them in the Q&A box. That just makes it easier for us to find them all at the end and ask our speakers. We have closed captions available on this webinar this evening. If you click the little CC box, they should appear on your screen. You should be able to make them bigger or smaller, and you should be able to move them around on your screen as well. I know sometimes when they're on the screen, they might be blocking something, so you can move them around. You can also hide them if you don't want them and they're just in the way. So this is the second in our series. We're looking at how cancer is researched in the lab. And after this week, we have two further talks, one about the ethics of machine learning and how it's used in cancer diagnosis. And then our final session in the series is a cancer research careers panel. So we'll be joined by 3 or 4 speakers and giving you an opportunity to find out more about different career pathways in cancer research. So all 4 of us in normal times would be based at the Wellcome Genome campus just south of Cambridge. I actually started during the pandemic, so I still haven't been to campus yet. So we're interested to see where in the world you are. So if you can find the chat box and if you want to let us know where you're from, we'd like to see whereabouts we've got audience in from this evening. So we've got London, Essex, Norfolk, Buckinghamshire, so got lots of east of England, Gloucestershire, India and Switzerland. I think that's the furthest so far. California or India. My geography is not good enough, so I think we'll say that you're tied for furthest away from campus tonight. And someone just down the road in Sawston. So welcome from near and far. Thank you for joining us this evening. On the campus then, everything that we do is to do with genomes. That's why it's in our name. And genomes has to do with DNA. We have 2 main research institutes. We've got the Wellcome Sanger Institute and we've got EBI. Wellcome Sanger focuses a lot on different areas of genomic research and has a lot of what we call wet lab stuff. So if you've seen scientists with a pipette doing stuff with chemicals, that's the sort of stuff that the Wellcome Sanger does. EBI is all data focused. So once we've done all our experiments, we have a lot of data that we can use to find out more about biology, and that's what EBI focuses on. We've got about 6 different areas of research that we look at. So we have human genetics, where we look at how diseases are passed down through families. We have cancer, which is obviously the area we're talking about tonight. Cellular genetics, so understanding how different cells operate. Parasites and microbes, which used to be mainly focused on malaria and right now has a big focus on COVID. Tree of Life, so looking at biodiversity in the UK, and biodata, so looking at all that data we get from our experiments. Before I hand over to our speakers for tonight, if anyone's doing GCSEs or A-levels or other sort of high school level learning, I'd recommend Your Genome. It's our online resource website full of articles, videos, and activities that you can do around lots of different topics, including cancer. And I'd recommend signing up for our education mailing list, which is where we announce new events like this and anything else that we're doing. So I'll put links for all of this in the chat box in just a second. But for now, I will stop sharing my screen and I will pass over to Luke to take over from here.
Good evening, everyone. It's very nice to speak to you today. So my name is Luke Harvey. I'm a MB PhD student at the University of Cambridge. So I'm 4 years into my clinical studies, and then I took 4 years out to start doing a PhD in cancer research. I'm doing it in the Martin Corona Lab in the Cancer Aging and Somatic Mutation department at the Sanger Institute. And so my talk today is on dissecting cancer evolution. So a brief history of cancer, and just to remind you all of what cancer is. So cancer is a collection of different diseases, all caused by abnormal cell growth. And what I mean by that is, so we have these kind of badly behaved cells that break the rules and they begin to grow uncontrollably. And we see diseases like this in all complex multicellular organisms. And they've affected us for as long as we have been humans and for as long as we have been anything except single cells. And so if you look on the screen, you should be able to see this picture of a bony tumor growing out from a leg. And this is from an Egyptian mummy from the 24th century BC. So even mummies had cancer. And the reason we call it cancer is because the Greek physician and philosopher Hippocrates described these large tumors he saw on skin that looked like crabs. So cancer means crab in Greek. And they looked like they were bulging, and the veins they had on them looked like a crab's leg holding onto the skin. So cancer as a disease is caused by our own cells that become mutated over time. And specifically, we're talking about somatic mutations. So this is the accumulation of DNA damage that happens in cells throughout our own lives. And this can happen from exogenous sources, so from outside of the cell, like UV light. These kind of high energy particles can cause DNA damage, or it can happen endogenously, so from within the cell, where we have double strand breaks occurring, where just accidentally the DNA can break, or we have metabolic stress from these like reactive oxygen species that are very high energy and combine to the DNA and cause damage. So a quick question to ask you then is, how many mutations do you think an average adult skin cell has? How much DNA damage would a cell accumulate during a lifetime? I think Em will put up a question.
So we've popped a poll up which should jump up on your screen. If you're having any difficulty seeing it, you can use the chat box to let us know what answer you think is right. So how many mutations do we think an average adult skin cell might have? So it looks like most people think it's quite a lot. So we've got nobody going for 0 to 10, a few people going for 10 to 100, and then a very even split between our top two sort of categories of 100 to 1,000 or 1,000 to 10,000. The highest one is just winning, but by like 2 votes.
Very good, everyone. Yeah, no, very nice. Yeah, so you're absolutely correct. So an average adult skin cell has between 1,000 and 10,000 mutations. So throughout our lifetime, these cells are accumulating thousands and thousands of mutations, but they're remaining healthy cells. This DNA damage, as I said, is coming from from UV light. And we'll learn throughout this talk about how DNA damage can vary between different tissues, and also why cells can have tens of thousands of mutations but remain healthy cells. So an important thing to know is that if we're having somatic mutations, that means there is diversity within our cells. If we take any 2 cells from our body, they will have different patterns of somatic mutations. And we can use that to tell how related 2 different cells are to each other. So in the same way that we share mutations with a whole array of monkeys, as you can see on this phylogenetic tree, all the cells within our body also share mutations with each other. In the case of monkeys, we have this common ancestor a few million years ago that we had a split into all the great apes. Whereas for the human phylogenetic tree of our blood, the most recent common ancestor is the zygote. So where the sperm fertilizes the egg, from that point onwards, all cells accumulate mutations and become gradually more diverse. So as I'm sure you've learned in GCSE and AS level biology, where you have genetic diversity, and if you add a selection pressure to it, you will have evolution. So within the body, the cells compete for space, they compete for food, and they compete against the body itself. The body has all these rules that control growth, and that creates competition. And so where we have diversity and the selection pressure, the cells can begin to evolve. And so if you look at this diagram on the left, you can see this population of stem cells. One of them becomes mutated, outcompetes the other, and a population grows. And then gradually over time, another mutation appears, more advantageous than the last. And we have these populations of evolving and mutating cells. And what they're evolving to do is they're evolving to grow more, so proliferate more and divide more frequently. They want to hide from the body's kind of growth suppressors, so the immune system and things like that. They want to grow their own blood supply so they can start getting their own food. And they want to become immortal and spread. And so these are the hallmarks of cancer, as we describe them, the kind of 6 things that all cancers want to achieve to grow. So which human cells can evolve to become cancer, though? Is it all cells, or just dividing cells, or just the stem cells we have in our body?
Again, we should have a poll on our screen. If anyone can't see it, pop an answer in the chat box. So it looks like most people are going for all cells, but there's quite a few votes for dividing cells and no votes for stem cells at the moment. The other two are flipping between which one's most popular. All cells is winning, but again, by about 4 votes.
Good. I've made the questions too easy then. So yeah, so all human cells have the potential to become cancerous. So we have brain cancers that develop from non-dividing neurons. We have skin cancers that develop from very rapidly dividing skin cells. So we'll look at how a bowel cancer can develop in a little bit more detail. So there are roughly 42,300 bowel cancer cases in the UK every year. That's nearly 110 every day. But how do these bowel cancers begin to develop from this healthy tissue? So bowel cancer is caused by the uncontrolled growth of colonic crypts. Now, colonic crypts, you can see down here in the bottom left, are the functional unit of the gut. So they increase the surface area of the gut by kind of growing and invaginating like this. And it's the site of absorption for all of those digestive juices and all of that food that we're eating. So we want to have the biggest surface area possible. What can happen over time is these cells can begin to mutate. So a very common mutation that occurs in bowel cancer is an APC mutation. This regulates cell division. And when this mutation occurs, these cells can start dividing a bit more frequently they should. And we get these things called adenomas forming, and you can see this irregular growth that's starting appearing on the lining of the colon. If they accumulate more mutations, so RAS is another cell cycle regulator, we can have these much larger, even more irregular adenomas forming. And eventually, through a whole range of series of further mutations, we have full-blown carcinomas forming with this very irregular invasive growth pattern you can see in the bottom right. But if you look at the ages of when these mutations are likely to occur, you can see that this can happen over a period of nearly 40 years, from 30 to 70, which is a very, very long time. And also, whilst I've said APC, RAS, and this series of genes here, cancers have a diverse range of genes that drive them. So how many genes do you think have been found under positive selection in cancers? So these are how many genes have been found to cause cancers?
So far people are going, again, for the high end of the scale. We've got a mix of responses from the 100 to 200, 200 to 300, and 300 to 400. Got a couple of guesses for 0 to 30, and no guesses for 30 to 100 at the moment. Yeah, it looks like pretty much everyone's gone for the top 2 categories, and 200 to 300 has won by 1 vote. They're very close at the top end.
They're finally wrong. So there are 300 to 400 genes. There's some debate over this, have been found under positive selection in cancer. So there are two enormous data sets that we've looked at, something called the Cancer Genome Atlas and the Pan-Cancer Analysis of Whole Genomes, that have looked at the genetics of thousands of cancers and looked for which cancers are most frequently mutated and driving cancers. And they find that there are nearly 400 genes that can cause cancer. But what's very interesting, so this is a rather horrible graphic, I'll admit, is that there are many different genes driving different cancers in different tissues. So what I want to draw your attention to is the two I've highlighted in red, colorectal cancer and pancreatic cancer. You can see P53 is frequently mutated in many cancers. You can actually, in fact, you can see it in the esophageal cancer, the head and neck cancer, and a variety of other cancers. However, there are some genes which are very specific to certain tissues, such as breast cancer, we have BRCA2 mutations, and some cancers, like colorectal cancer, has many, many genes driving it, or bladder cancer. But some cancers, like thymus cancer, there are only 4 genes seen mutating it. So there's an enormous diversity in cancer driver genes between tissues and within cancers. So the question is what drives this diversity of cancer mutation? And how can we study this evolutionary process? And to do this, we need to identify how DNA damage accumulates in healthy, normal cells. We need to understand what mutations are okay and which mutations are bad. So how can we design a study to research normal cells? So what we need to research is cells at this stage. We need to research when they look healthy and normal, and we need to research that in thousands of patients, and we need thousands of samples to do that. And so at the Sanger Institute, we've developed this pathway where we can collect tissue samples from transplant donors. So when people have passed away and they leave their organs, we can collect these organs. Routine screening, so things like cervical smears, colonoscopies, things like this, and surgical waste material. So when people have joint replacements, or when they have any kind of plastic surgery and there's material left over, we'll happily collect that material. And then what we do is we embed this material and make slides from it. So I'm sure in your biology labs, you've made slides of onion cells probably, but we do a similar thing, but with kind of human tissue. And then we stain these cells to look with hematoxylin so that we can see the DNA, eosins so that we can see the proteins in the cytoplasm, and fluorescent markers so that we can see specific genes and specific cell types. And what will focus my talk on for the rest now is this process we've developed called laser capture microscopy. So what we see down the microscope after we do this staining process is what you see on the screen in front of you. So on the left is healthy colonic crypts from the colon of a mouse. So you can see the folds of the gut increasing the surface area, and then you can see these little worms, as they look like, which are the colonic crypts along the colon. And on the right is a section of a human colonic crypt. The issue is that parts of what the cells that we're interested in are just these cells that kind of form these villus structures, the colonic crypts. But surrounding it, you can see all this muscle material, all these lymphocytes and material we don't want. So the question is how can we isolate those cells of interest? So we use laser capture microdissection, or the LCM as we call it. And what this is on the left, you can see a picture of the LCM we have in our lab. It is a very high-powered photon laser, which is attached to our microscope. So we can isolate material in the same way that you would play a computer game, you would control it with your mouse. We control this laser with our mouse as we look down the microscope, and we can cut out those cells of interest. So on the right now, you should be able to see a video of this laser taking place. So this is me sat at our computer in our lab, and I'm just clicking and dragging this laser with my mouse. It's very, very simple for something which I think can effectively cut through steel, but we use it to cut through small kind of collections of cells. And so what I'm doing now is I've cut around it, and that is going to fall out, and we'll collect it in a little well, and we'll send that for DNA sequencing. And so we've done this on thousands of individuals. at this point. But what I want to talk to you today is about one of these studies we did in the human colon. This is by my colleagues Henry Lee Six and Sergej Olofsson, and they dissected 2,500 individual colonic crypts from 88 individuals. So these images aren't as nice as the ones I showed you before, but you can see this, if you look in the bottom left, you can see this kind of plane of colonic crypts, these healthy colon cells. And what we do is we dissect them out. So then you can see that colonic crypt falling out. And then if you look at the image on the right, this is that plane of colon and all the colonic crypts cut out. And we send those for DNA sequencing. And what we want to know is how many mutations are in a normal colonic crypt, what genes are mutated, and can we find evidence of evolution taking place? So what we found out is in a 50-year-old man, there's roughly 2,500 mutations in each colonic crypt. Driver gene mutations were very, very rare. In less than 1% of the cells did we see any sign of a cancer driver appearing. And clonal expansions, these signs of evolution, were equally rare. We didn't really see them occurring in the colon. What we can do is, if you look on the right, this family tree that we made of the colonic crypts, we can see how closely related they are to each other. And where we do see a driver gene in this axon 2, which is these 2 red dots, they're very, very closely related, suggesting that there was a mutation at some time point quite frequently. And these cells are beginning to expand and spread throughout the colon. And this could potentially be the beginning of cancer development. So we've done this similarly in skin. So this material was collected from someone having eyelid surgery. So this is someone's eyelid preserved. It's not a slug. And what we did with this is we cut out lots and lots of little circles. So rather than with the colon, we had to dissect and isolate these kind of rare cells from the background. In this tissue, we just had a sheet of skin cells, so we could just cut circles out of them. And as I said before, skin cells mutate lots and lots because they're exposed to UV light. Some of these cells had well over 10,000 mutations. What was very interesting is these healthy skin cells were mutated with cancer drivers very, very frequently. So P53, that gene you've probably heard of as the guardian of the genome, was actually seen mutated in around 10% of all healthy cells in the skin. And Notch1, this other famous cancer driver, was found mutated in 25% of the healthy cells we looked at. And this image on the right is showing 1 millimeter squared of skin and the size of these clones that we saw in the skin. So if you looked in figure B at one of these larger circles, this figure of this kind of balloon-like figure of lots of circles is showing all the related cells within that patch. And you can see somehow, for some reason that we're not quite sure yet, these cells have multiple cancer driver mutations and yet are remaining completely healthy. So there is a lot more research that needs to be taken place in this evolutionary process of going from healthy cells to cancer. Lastly, the work that we're doing now is we want to look at tissues that have lots of different cancers developing from them. So in this instance, we're looking at the pancreas, and this is a lobe of the pancreas. I'm sure you've learned about it in biology. The pancreas produces your digestive enzymes and it produces insulin. And 3 unique cancers can develop from the pancreas. From the acini, which produces the kind of digestive enzymes, we can have acinar carcinoma. They are in green. All of these kind of white lines highlight and surround the acini. So most of the pancreas is acini. The islets are in blue. I've highlighted one down here. They're quite hard to see, but they're collections of kind of quite dense nuclei, and they produce your insulin. And then the ducts, which drain these digestive enzymes, you can see in black. And so what we can do with the laser capture microdissection is we can cut out every single acini we see, every single duct we see, and every single islet we can see. And we can start to identify how from one tissue, multiple different cancers can develop. And so within our group in Sanger, what we're beginning to do is to dissect all human tissues. So I think we've now at a point where we've sequenced nearly 1,000 individuals. We've sequenced tens of thousands of samples from these individuals, and we've sequenced nearly every organ from within the human body. And what we really want to understand is how mutations accumulate in different tissues, how cancer drivers accumulate in different tissues, and understand this cancer evolutionary process. So hopefully what we've learned today is that human bodies are kind of these molecular mosaics. There's an enormous amount of diversity within cells and within our body. Mutations occur frequently in healthy cells. On average, across all tissues, there's roughly 1,500 mutations per cell at 50 years. Driver genes vary between tissues. They're very rare in the colon and they occur at high frequency. I've written esophagus here. I meant to write skin. And types of DNA damage can vary massively between tissues. For example, with UV damage in the skin. So if any of you have any questions, I'd be very grateful to answer them. And thank you very much for listening.
Awesome, thanks so much for that, Luke. There's a couple of questions, one that I think Patricia might touch on, so I'm going to leave for just now. But there's a couple that are quite specific for what you talked about. So one that asked around how the sort of genes and sort of cancer genes work. So is it bad to have the normal version of the gene, or is it only bad to have the mutated version of a gene?
So that's a very good question. So if we go back to this skin slide, so I remember speaking to Sir Mike Stratton, who is the director of the Cancer Institute, and he was saying 10 years ago when they first sequenced the cancer genome, what they were expecting to find was in a cancer, you would have one driver gene that was mutated, and that would be causing all of that aberrant cell growth. And then after they sequenced that first cancer genome, they saw there were thousands of mutations across lots and lots of different genes. And it was a very complex picture. And then later, they started sequencing normal tissues like skin, and they found that cancer genes were mutated very frequently in cells that were remaining completely healthy, weren't going crazy, weren't expanding, weren't causing any disease. And so the answer is it can be completely fine to have cancer driver genes and cells can remain healthy. The step that we're trying to focus on and trying to learn about is what the switch is from going to being a healthy cell with cancer drivers, but still having your growth controlled and not going crazy, to going to a cell with similar mutations that is all of a sudden cancerous and is spreading and causing disease in individuals.
Awesome. I think that's a really interesting point, and someone asked a similar question, but I think you've probably just answered that. The other question that was very specific to what you were talking about, someone's asked whether you can use the LCM, the laser method, for prognostic studies, and how feasible would that be?
So that's what we're trying to develop at the moment, is a technique of using this to kind of study and identify disease before it happens. Part of that was slightly complicated from these findings in the skin, where we found that DNA mutations and driver genes could occur in healthy cells. The other issue is, if we look at these pictures of the colon, so let's say we cut out 1,000 colonic crypts from an individual. So actually, that would be, I don't know, a few square millimeters of colon, where the colon itself is nearly 10,000 times the size. So the LCM probably isn't the best tool for kind of having an idea of what's happening in the entire gut. We have to kind of restrict ourselves to little regions of it, and we can randomly sample. But what we can try and do is, once we have an idea of what mutations are good and what mutations are bad, we can take samples from like poo, and we can see, are there any of these mutated genes in that sample? Because that's gone down the entire gut. And so it's sampling every single colonic crypt. And we can see, is there any sign in this patient that there is cancer? If there is, then we can begin to look in even more specific examples, and we can do a colonoscopy and try and take biopsies. And then if we wanted to, we could even do LCM and try and dissect out regions that we think are healthy and cancerous and look at those differences.
Awesome. That was really great. There are a couple more questions, but a lot of them I think Patricia is going to touch on. So thank you very much, Luke. And Luke will be answering more questions sort of at the end after Patricia's talked as well. But for now, I will pass over to Patricia to talk about how we might develop some drugs for these cancers.
Hello, everyone. I hope you can see my presentation. I'm going to talk about how we use cancer models to look at drug combinations and which drug combinations might work in patients. And cancer models are surrogate patients. So we can't obviously do experiments on the patients themselves. So we take their tumors, turn them into models to be grown on plastic, and then we can use them to do our experiments. And with that, I'd like to ping back a question to you straight away is, how many cancer models do you think are available to cancer researchers around the world? As a reference, we have roughly 17 million new cancer cases every year. But you can see already from the answers, we don't have as many cancer models. So what do you think, how many do we have available roughly?
It's very split across all four groups. Yeah, it's constantly changing. I think the 2,000 to 3,000 is winning by 2 right now, but it's between the around 1,000 and the 2,000 to 3,000, but very, very split.
Okay, that's very, very good to hear, because that is roughly what I would estimate as well. And why I say I would estimate that, it's almost impossible to know how many cancer models there are, because many labs have their own and they might not share them with the community. So we don't really know how many there are in total. But I think a good estimate of what a cancer lab might buy from a repository, for example, would be like 2,000 to 3,000 different models. And we're working on generating more and more. And that is what I'm going to show you slightly later on as well, how we do that. But let me talk about our lab and how we set up our things. So this is an assay that you can see. We have a viability assay where we want to see, are cells still alive after we treated them with drugs? And to evaluate the drug combination, you have to treat the cells first with drug 1 and then with drug 2, and then you treat them with 2 drugs together and you compare that. So what we want to see is, in a good combination, it kills more cells than the single agents. And this is one of our actual screens that we have done. It's a really big one. So I have been working on this type of data for 4 years now, and it's coming to a conclusion. So I'd like to take you a little bit on a journey today of how we generated the data and how we look at the data afterwards. So first of all, it's all about choosing which cancer models you want to look at. So it's mostly around which tissue you want to look at. We decided to go for breast, colorectal, and pancreas cancer, for which we have quite a lot of cancer models available, so 128 in total. For other tissues, we have very, very few available. But for those 3, we have a good amount for each of them. Then we chose our drug combinations, which in this case was over 2,000 in total. What we do with them is we chuck all those drugs onto the cancer models, and we evaluate how alive the cells are after 72 hours. And from that, you can calculate how much you think should have died when you look at the single agents, and then you can look at how many actually died and whether that is better than you would have expected. Another very, very important point is you have to know your models. So you have to know about which mutations are present in those models, whether certain genes are overexpressed or underexpressed, and what clinical subtype they represent. For example, breast has very good clinical subtyping. You might have heard of triple negative breast cancer, for example. So we know for all our models within the breast, for example, whether they are triple negative or not. And that we match up. So we match up drug responses with the characteristics of the models themselves. And that is how we find combinations that we think are valuable. And then we take them back into the lab and we really check whether the drug effects that we have seen in the big screen hold true. And for that, we do a lot more experiments. And then we take all that knowledge together to hopefully bring it to a conclusion for patients. So we want to know what drug combination would work for which type of cancer patient. So it might be a certain subgroup of pancreas patients whose tumors would react really well to a certain combination, but other pancreas cancer patients, their tumors might not react at all. So we try to understand that. And with that, I would like to show you how we generate new cancer models. And that is, it's a lot of work and a lot of hope. So what we get in the lab is we get small pieces from surgeries. So when the surgeon removes a tumor, they might cut a little piece for us, send it to us to the lab, and then we process it. We cut it up and we basically put it on plastic for cancer cells or in a matrix for organoids, which are two different types of models, as you can see in the schematic. And then we just hope that they grow. It's very basic. This is how it looks like. I hope it's not too gruesome for you. There's no real blood. On the left, you see how the piece arrives with us. It's an ovarian cancer sample. And then you can see that it has to be cut into small pieces to then hopefully grow. So that is really just helping the cells to split up a little bit more. We put them into media so that those are liquids with all the nutrients in it. You can think of it as a smoothie that contains everything you would eat in a day. So that is what we put on the cells, and we give them as much as they need. And every cancer cell or every cancer model might have their own requirements. So breast models might need slightly different nutrients than colon models, and so on and so forth. As I said, we put them either on plastic or in matrices, depending on what type of model we want to grow. And then we really just cross our fingers and hope they grow and don't die. And that can take weeks and is often quite unsuccessful. But if you have it right of which type of nutrients they need, the success rate is fairly high. So we can generate models this way.
We just had a really nice question on whether you need the patient's permission or whether the patient knows whether this is going to happen with their sample.
Yes, so ethics, it's all about the ethics. We can't do anything without the permission of a patient. It can come in various ways. So it depends on the hospital that the sample is collected in, how that actual agreement looks like. So it could be a fairly general agreement that the patient goes through with their clinician and then signs on, yes, my samples can be used in research. Some of them are way more specific, so they can be used for this type of research, but not for that type of research. But they always have to give their permission these days. Very good question, and very, very tricky.
Awesome, thank you for that.
Arrange that. What you can see here is the difference of how they look like in culture. So if you look through a microscope, on the left, you can see a cell line. On the right, you can see an organoid. Both of them are derived from pancreas, but you can see they look fairly different. So on the left, the cell line is 2D. It grows in one layer, so quite regularly. You can see that the cells are one next to the other, and they are adherent. So they grow attached to plastic. It's treated plastic, so not normal plastic like you would have it at home, but it's still plastic. On the right-hand side, you can see organoids. So those are cells that grow in complex structures that are 3D. So the bubbles, in a way, that you see are cells next to each other. And because of the way the microscope works, you don't really see that it is 3D, so it cuts a plane through. But that is how the cells are arranged. So they are in 3D structures, and often the bubbles might not have cells in the middle, but some do. All the tissue types look slightly different. Very important, they are not adherent, and that is because we grow them in matrix, as you can see at the bottom. So those blobs is a matrix of different factors that we grow the cells in, and they keep them 3D. And with this, I would like to ask you an open question. What do you think might be, so please pop your answers in the chat box and Emma's going to read them out to us. So what do you think might be advantages of using organoids over cancer cell lines, for example, in cancer research?
So you've got any ideas, having a think about what Patricia just said about the difference between the two, pop them in the chat box for us. We'll give you a couple seconds to have a think.
Just to encourage you to answer, I think the research community does not necessarily know the answer. So there's no right or wrong that you can answer. I think it's all about different ideas, and people are working on finding out what are the real advantages and what might not be a real advantage, even though we think it might be one.
Got a couple of really nice ideas. So one that suggests that the sort of normal cell lines might be a bit too simplistic. A couple about sort of accuracy in terms of what it's really like in the body. So maybe to stimulate the organ environment, or yeah, just a more accurate representation of what it would really like be in the body. Someone's put to watch the progressions of the mutations within the sort of organoid. One idea about having a control variable, which I think is really interesting because that's obviously really important in science. Someone's put to see the cells in proper relation to each other. So yeah, lots of ideas about it maybe being a bit more what it's like in the actual body.
Yeah, I completely agree. I think that is the most obvious point that we think really makes a difference, that they are in structures. So they interact with each other different than they would if they are grown in one layer on plastic. So it looks a lot more like the architecture the tissue might have in the body itself. And I completely agree that is one of the most important points we think makes a difference. So at the moment in our lab as well, we are treating models from the same tissue, so cell lines and organoids, with different drugs. And we try to understand whether it makes a difference in drug response, for example, whether it is in a 3D structure or in 2D. So that is definitely a very important one. One that might not be that obvious to you, that I didn't know of for a long time, is that even though we have a lot of cancer cell lines available, and that 2,000 to 3,000 is mainly cancer cell lines, not every cancer type is really represented. So some of them cannot really be grown on plastic. And there we try to grow them as organoids, and we have success with some of them. So some of them really don't like to be grown in 2D. So that is where we can fill gaps in patient representation in our models. So that is certainly an important point as well. But in the end, what I think will come out is that both of those types of models are quite complementary, and we might be able to answer different types of questions with each of them, or we can potentially just pool them for a certain tissue type. So use the two different types for the tissue and then answer the questions using them together. So with this, I would like to show you a little bit of how it looks like in the lab. So on the left-hand side, you see a cell line grown on plastic, so in a flask, where the medium, that is this light orange that you might be able to see, is in, and then you look through the microscope and that is how it looks like. We always have to check ourselves before we do any experiment with them to check that they are healthy in the beginning, because you don't want to be working with something that is not that right in the beginning anyways. Then in the middle, you see a cell culture hood, or two hoods actually, sterile environments where we manipulate the cells, so where we might add the drugs, for example. And then on the right, you see cell incubators, which keep the cells at 37 degrees at very controlled oxygen and carbon dioxide environment, so they are happy to grow. Then this is a little bit about how we choose the drugs for our screen. And that is a lot of work and a lot of thought goes into that. So you want to be sure that you are as sure as you can be to use the right drugs for the right tissue, for example. So in general, we focus on compounds that are commercially available, simply to make them available to us. So we have to buy them in. And then we consider quite a lot of things that are tissue specific. So which drugs might be used for colon cancer in clinics, which are already in clinical trials? Have there been any reports in literature, what might work, what might not work? Are there any drug combinations that are already known in that cancer that we could use as a positive control? So these are all considerations around the tissue type. And then if we still have space in our screen, we will cover as many biological pathways as possible, just to give us an advantage in like exploring as much space in the combination world as we can. So for me in practice, I was reading a lot of literature before we start a screen, and it's still very scary because you're afraid that you missed something obvious, but you have to do your best and then you just go with it. And on the right-hand side, you see one of my favorite robots in the lab, she's called Charlotte. And that is one of the robots that we use to prepare our drug stocks. So to get the drugs at the right concentration, we use this robot to dilute the drugs from when we buy them in. This is how a drug plate layout looks like. It has 1,536 wells. So in each well, you will have cells, and then you can add a drug on top. What you can see is that we have a lot of controls. So we have positive and negative controls, and the negative controls, the cells should be happy, and the positive controls, they should die. So we use that to normalize our drug responses to understand whether our drugs worked or not. And then in green and blue, you see the drug combinations here screened as a matrix. So 7 concentrations of one drug, 7 concentrations of the other drug screened against each other. You can see that we have 15 different combinations on this plate, which is really magnificent and you would never do by hand. So we have robots who pipette all that and who are way more accurate than a human being could ever be. This is how it looks like after we have drug treated the cells and waited for 72 hours, and then we read off the viability. What you can see is viability is the highest when it is yellow, and the cells died where it is red. If you see like certain red spots all over the place, those are the positive controls where they should have died. And then if you see clusters of red, like bigger ones, this is where a combination was added to the cells that then killed the cells. So this is how the raw material, the raw numbers look like. We take those raw signals, we normalize them to the positive and negative controls to get like, this is how a happy cell looks like, this is how a dead cell looks like. And then we get all these metrics of them. So for the single agents, for the combinations, lots and lots of numbers. That is how I see it. And we get very, very big tables of numbers. And that is actually so much data that when I got my first data file back, I couldn't open it in Excel. I think everyone who's used to Excel will be with me. It's like, oh my God, what do I do? So this is what people working with big data are facing. They have to learn programming tools like R or Python to look at the data. Actually, that is a very steep learning curve for someone like me who is a trained lab scientist, but I've become to love it. So I love computation biology, and for anyone who's interested in dealing with numbers, please look into that and look into programming because it might be quite fulfilling. So what I do in my day-to-day job is I explore all the data that we have. I look for general trends. So we have 3 different tissues, breast, colon, pancreas. Is perhaps one of them more sensitive than another one? That might be a general trend to look for. But then I really want to understand which of all those 2,000 drug combinations works the best and why does it work really well? And that is where I think, well, I've gotten to a point where I think, well, I found a combination that works. But it's like a needle in a haystack. You find it and then you doubt yourself. It's like, okay, I have done a big screen, but does it really work? And that is where what I mentioned, the validation work becomes really important. So we perform a lot more experiments to understand whether the effects that we see are true or not before we ever propose that they would go into a patient. And these experiments can come in our variety of experiments. In blue are the ones that I would rather call proof of concept. In green are the ones that are more clinically relevant. So when I go back into the lab, looking at a combination in detail, I'll look at, first of all, again, a viability assay. So the one that we have done on the big screen, just confirming what we have seen before. Then I'll be looking at whether the cells actually die or whether they just grow slower, because those are 2 different things. I would try to understand whether it's really just one of the 2 drugs driving the effect or both of the 2 drugs. What is the mechanism behind? Do the 2 drugs really act together, or like really just understanding the biology behind? If we had drugs that target more than one target, and they often do, so they might inhibit 2 different proteins, we have to understand which of those 2 was the driver, like which is the more important one. And then for the clinical considerations, it's really understanding which patient would benefit. And that is where we go back to knowing as much as we can about our cancer models. So for example, we would want to know whether a combination that works in colon cancer models, whether it works in KRAS mutant colon cancer or in KRAS wild type cancer. And that is the drivers that Luke mentioned. So KRAS is a driver mutation in colon. And that is really we have to select our patients well. There's no point in treating a patient who might not benefit from a drug. And then with drug combinations, you have all kinds of other considerations. So how would you dose 2 drugs? Would you, like, which concentrations would you give to a patient? Would you give them on the same day? How long would you give them for? All those questions around dosing. And then very, very important, dealing with the side effects. So single agents can be toxic and can have quite severe side effects in patients. You can imagine that when you combine 2, it gets incredibly more difficult. And that is, you have to do as much as you can with your treatment to evaluate all those things before you ever take it into a patient. So that is what we try to cover when we do validation work. And with this, I would like to come to my personal conclusion. Lab work is super exciting. I love it. I love working in the lab, but it can be very repetitive. So setting up a screen, like growing all the cells, is a lot of repetitive, slightly boring work. And it can be very frustrating at times when you do an assay and you try something new and it doesn't work. And you try it again and it doesn't work either. And you might have to try something 5, 6 times until you can make it work. And that can be quite frustrating. So that is for anyone considering going into the direction of doing some lab work. It is usually exciting, but there are some dry patches on the way. So please don't give up and boost your resilience. And with this, I would like to thank all those people who are involved. And you might imagine such big data generation projects. It involves a lot of people. So we have a fantastic team in the lab who do the big screens, 7, 8 people slaving away for, I would say, for this one project that I showed you, a year. So that was a year's worth of work of 8 people just to generate the data. Then it was another 3 years for me looking at the data, and another year for me and students and others going back into the lab, evaluating and doing more experiments. And then you have in between all those people, IT people who help us deal with the data, collaborators and other institutes who do parts of the work. So it's a collaborative effort, but I really enjoy working in a team. And thank you. If you have any questions, please go ahead.
Awesome. There's a couple of good ones that are specifically about your work, Patricia, and then some more generally that I'll let either of you jump in, depending on who would rather. One asked, why do you wait 72 hours after drug treatment? Is the 3 days, has that been chosen? Is it sort of a random pick?
Yeah, it's, I don't want to say fairly random. It's based on what we can do in the lab. So you want to treat cells that are happy, which means that when you seed them and when you put them down on plastic into those plates, you have to seed a certain number of cells. And you don't want to seed too many because then they might overgrow if they're happy, but you don't want to seed too little because then they might be too sensitive to the drugs. So dealing with a lot of cancer models, I think 72 hours is really just a compromise between all those factors. Other labs work with 48 hours, others do 96 hours, but that is roughly the ballpark that we're in. It's always between 48 and 96 hours that we generally do our experiments in.
So many practicality rather than anything else. Someone's asked whether you can use organolds to help work towards personalized cancer treatments.
Yeah, so that is, I mean, that would be my perfect idea of how to treat a patient, is not only knowing about the tumor. So what is being done now is that tumors from patients are being sequenced on the spot, more or less. And then treatment can be tailored towards how, like which mutations a patient's tumor has, for example. But it would be even more ideal, I want to think, to generate a model and then take it back to the lab, treat it with different drugs and see real time which drugs the tumor would respond to. Unfortunately, we're not there yet. And I think the biggest drawback at the moment is that generating an organoid takes months before you can do any assays. And that is time that the patient doesn't have. So while we hope that in the future that might be possible, whether with organoids or a different type of model system, we're not there yet.
Awesome. And then, yeah, a good couple of questions that were more generally about sort of both of your work and cancer more generally. Someone's asked why, it's a really nice phrasing, why would nature give these cancer gene drivers to us? Is there any advantage of having sort of cancer mutations or mutations in general?
I've been thinking of an answer for it. So we have humans have 20,000 coding genes, and as I said, there's roughly 400 of them which can become cancer drivers. And the types of genes that become cancer drivers are all involved in regulating how cells divide, how DNA is repaired, and how DNA is synthesized, things like this. So for an example, if you remember on my slide, we had that RAS protein that was mutated in bowel cancer development. And RAS is basically an on-off protein, works as an on-off switch that tells cells when to divide correctly or not divide correctly. So it's normally on when a cell is going through the division process, and the rest of the time it should be off. Now, if we have a mutation that affects one of these kind of enzymatic sites in the RAS protein, it can be switched permanently on, and that cell can divide constantly. Another protein which is talked about quite a lot is P53. So P53 is known as the guardian of the genome, and the reason it is because it basically works as a detection method for DNA damage. So when DNA damage is present, there's a whole array of different proteins that signal to P53 and say, hey, we have a problem here. We need to stop cell division and repair DNA damage. So then when P53 is mutated and it can't do this signaling process, suddenly DNA damage is allowed to accumulate at a much higher rate than it was before. So cancers with P53 mutation normally have very high mutation burdens, often in the order of hundreds of thousands of mutations, compared to healthy cells, which just have a few thousand. So all the cells that are genes that are cancer drivers have very important roles in regulating normal cell division and normal DNA repair. It's just they have the potential to become oncogenes and drive cancers if they're mutated in the right way.
Awesome. So we need these genes in the first place. It's just that sometimes they can go a bit haywire. One that I think nicely links together both of your work asked about how we can sort of prevent mutations from happening in the first place, whether we can design drugs to prevent them. Might be something that both of you might have ideas on.
Well, that is a very interesting idea. I would rather say stay off the drugs to prevent mutations, to be honest. So we know about some environmental factors that can foster mutations. So alcohol, like overuse of alcohol or tobacco, for example. But even then, even if you follow all the rules, you might have mutations popping up, and some people are more susceptible than others. So yeah, it's all about prevention, but how to do that beyond some general recommendations is very hard. So there is no drug, unfortunately, that can prevent mutations these days.
Yeah, sun cream is good.
Yeah.
Sun cream is the best drug for anti-cancer. You both talked a bit about P53. So someone's asked whether it's possible to design a drug that only targets cells with a mutated P53.
Again, that is very, very interesting. So generally, the drugs will go to every cell and will target whatever they were designed to target. But there is a lot of work on delivering the drug to the right cell, so only to the cancer cells, for example. And there has been quite a lot of hype in the last decade, I want to say, around certain ideas, but not many of them have come far. And I don't fully understand why. I've never really worked on those topics, to be honest. But yeah, I mean, there are a lot of ideas out there that, for example, a drug is only activated in the cancer cell. So if it encounters a non-cancer cell, it might not be active, it might not do any damage. So there are a lot of ideas, and certainly that is a very good concept and one for the future. And I hope that it will come to fruition and get better.
Yeah, just add to that, we're just really bad at making drugs, to be honest.
Yeah.
If you look at the FDA in America, which approves drugs, I think in 2018 they approved 20 new drugs across all diseases, and on average it's less than 50 they approve a year. But we're trying, every year there's thousands and thousands of drugs in development, and so if we want to make drugs that can target p53 and target these driver genes, we're just not very good at it. Most of the drugs that we do use in cancer treatment were discovered accidentally, or they're repurposed from other things. It's a slow process, but certainly in the last few years, and with things like organoid development and very good cell lines, we are definitely getting much, much better at making good drugs to target specific genes and proteins.
Awesome, thank you. I'm aware that we're coming right up to past, so I know that there were a few more questions in the Q&A. So if anyone's got further questions, you can email us, and I'll pop our email address in the chat box in a minute. And if you want further answers, just pop them through on the email. But thank you very much, everyone, for coming along, and a special thank you, obviously, to Patricia and Luke for joining us this evening. Our next session next week, following in our cancer series, we'll be looking at the ethics of machine learning in cancer. So a really interesting area of using AI and machine learning to help diagnose and spot cancer in a very different way from how we were talking about spotting cancer today. So I'll pop some links in the chat box in a second, but yes, thank you everyone for coming along, and hopefully we'll see you again next week.
Thank you all for listening.
Guys.